what we discussed, what we learned. Aisha, are you there? It's not a, don't take these summaries as a punishment, okay? Just take it as a opportunity to just start the class. So you start the discussion, you ignite the fire.
So,
Yes, Iram, you would like
so
to do
if
that?
she doesn't want to, she can offer it to me.
Yes, Aisha has some throat, still her throat is also recovering. Okay, so yes, Iram, would you like to do it?
Yeah, sure.
Okay, great. Thank you. Go ahead.
So in the last class, we talked about salient features of double helical structure of DNA given by
Mhm.
Watson and Crick. First,
And sorry to interrupt you, Iram, but sorry to interrupt you. But how many of you know the story of Watson and Crick or anything about it? Like even a little bit more than what textbook says. Sorry, I just lost electricity, but I hope you can see my very bright smile. It will come back. Don't worry. Yeah. Iram, do you know anything about Watson Crick? Because we are talking about what this is the science that they gave to us. You know, where did they figured out in their head that a helical structure can define DNA? You know, where did that knowledge came to their head? Where were they exactly when this thought came to their head? And it happens with most of the scientists who are amazing and who are scientists by passion. They're not scientists, they're just thinkers. Okay. So in earlier times, when there was no degree, PhD kind of setups, we used to call these people philosophers. So in every culture, religion, race, and part of the world, we have had philosophers in the past. Then those philosophers started schools or universities. So school simply means a group of people who believe in certain ideas and then they talk about it. That's called a school. In science also, for every topic, there can be multiple school of thoughts. For example, when people didn't know about DNA structure, there were many groups around the world who used to think, oh, DNA could be like this. DNA could be a ringed molecule. DNA could have all the double bonds and a carbon backbone. Then someone said, no, DNA, I think, seems to have something more acidic. So there are many schools of thoughts about DNA. They were all logical thinkers and philosophers. So Watson and Crick, very interestingly, came about this idea. They were working on it, and they were actually in pub enjoying with friends. And there's a very beautiful book. Just give me 1 second. I want to show that to you to inspire and encourage some. Just give me 1 second. Let me figure it out. I have to put my screen down. You can still hear me, right?
Yes, sir.
yes, sir.
Everyone. Okay, so yeah, I found it. So that's the classic thing. And it's a real original copy. And I cannot resist showing it to you. So this is a book written by James D. Watson. And it says, so this book is titled Genes, Girls, and Gamos. And it says after the double helix. So what happened once they figured out or they got this idea that DNA could be a double helix? So it says that in the years following Watson and Francis Crick discovering the DNA, James Watson was obsessed with finding two things: RNA, and all these discussions used to happen in pubs, okay, with friends. So Watson find two things through this discovery: RNA and a wife. Okay, so that's where the name comes: genes, girls, and gamos, because through this discussion he found his life partner. So Genes, Girls, and Gamo is a marvelous chronicle of those pursuits, and Watson has written this book very, very beautifully. So if any one of you is interested in reading how, so when we imagine scientists, we imagine some madmen wearing white cloaks, with dark circles all around, with some fancy things in hand and purple and red gloves that looks like some villain's hand, and they're doing something in the lab. But that's not the exact true picture, right? I aspire to become a scientist in my life. So I take inspiration from people like Watson and others. So do find this book. There are PDFs also available, and try to read some chapters. It's super interesting and fun. Yes, Iram. So what we were talking about, Watson and Crick's.
Yes, sir.
Yeah.
So we were talking about salient features of double helical structure of DNA given by Watson and Crick. First, it is made up of two polynucleotide chains, and the backbone is constituted by sugar phosphate.
Yes.
And also, the bases project inside. The second point is the two chains have opposite polarity or they are anti-parallel. It means if one strand has 5' to 3' polarity, the other will have 3' to 5' polarity, just the opposite.
Yeah. And how do we decide what is 3' and what is 5'? Which side we will call 3' and which side we will call 5'. Remember, I told you that whichever side the phosphate is towards, just like how we decide in east and west. East is from where sun rises. So east is where the phosphate will be in that nucleotide. So if you take one nucleotide of a DNA, and if towards the left-hand side, like you can see here on the screen, the phosphate group is towards the left-hand side. So the left-hand side becomes the 5' because there is a 5' phosphate. And the right-hand side becomes the 3' because of a hydroxyl group present in that sugar, the pentose sugar.
Yes.
Yeah.
Yes. Okay.
Yeah.
The third thing is that the bases are paired through hydrogen bond or called as H bonds, forming base pair. Adenine will form two hydrogen bond with thymine, and guanine will form three hydrogen bond with cytosine. Then we
Right.
discussed a question why a purine always comes opposite to a pyrimidine in a DNA double helical structure. A purine always pairs with pyrimidine to keep the distance between two strands of the DNA uniform so that a double helix is formed.
Correct.
Then the fourth point is that the two chains of the DNA are coiled in right-handed fashion. There are three types of DNA: bDNA, zDNA, and aDNA. bDNA is common in human; it's the biological DNA.
Yeah, it's common in biology everywhere. Okay, it's the most like almost all organisms under natural conditions have bDNA. Okay,
Yes,
Z
sir.
and aDNA are rare, but they are alternate forms of DNA. DNA can also form improperly to become the zDNA.
Yeah.
Yeah.
The pitch of the DNA, which is one turn, is 3.4 nanometer, and there are 10 base pair in each pitch, which means that the distance between two consecutive base pair is 0.34 nanometer. Okay.
Yeah.
Then the plane of one base pair stacks over the other in a double helix, and this provides stability to the helical structure. And this is also the reason why DNA is more stable than RNA.
Yes. One of the reasons.
Uh,
Yes. Great.
yeah. Then we talked about euchromatin and heterochromatin. Euchromatin is the part of the gene which is activated, and that gene is called as euchromatized. And the gene which is silent is called as the heterochromatin DNA. Then the length of DNA in a haploid genome is 1.1 meter, and the length of DNA in a diploid genome is 2.2 meters. Yeah. Then we talked about packaging of DNA. In DNA, the proteins required for packaging are DNA binding histone protein. DNA is a negatively charged molecule because it is an acidic molecule. And if DNA is acidic, then the proteins required will be positively charged to make it compactly packed. Yeah.
Because
And
if you give a negatively charged protein to a negatively charged DNA, they will both repel each other. Because
yeah,
in chemistry and physics, you have studied that like poles or like charges repel each other, right?
yes.
Yeah.
Then in prokaryotes, the packaging is done by a similar positively charged protein that holds the DNA in a dense region. It's called as nucleoid. In eukaryotes, the organization of DNA is much more complex, and it's done by positively charged basic proteins called as histone. So
Right.
this histone, it is made up of 8 single units. This is called as histone octamer, and DNA is wrapped around it. Then there is also H1 histone that clips the DNA molecule. Histone octamer is also called as core histone protein. And this whole thing is nucleosome assembly. Yeah.
Yeah. And
Nucleosome assembly. Yeah.
one thing I forgot to tell you because it was not in the book, but then I realized I should have told you is that histone octamer. So H1, so there are many histones, right? Histone that makes this clipper, histones that make the octamer. So different histones have different names. The H1 is present in this last clipper molecule, which is again positively charged. The histone octamer is made up of H2A, H2B, and H3 histones. Okay, so these make the octamer. Is it clear?
Yes, sir.
Yeah.
Okay. Then 200 base pair are bound around a nucleosome. So one nucleosome can wrap 200 base pair of DNA. Then all
Yeah.
the nucleosome together form chromatin, which is called as beads on string-like structure. Then chromatin is further packed by non-histone chromosomal protein, which is NHC protein. This forms euchromatin or heterochromatin also. If it is densely packed, then it is heterochromatin, and if it's loosely packed, then it is euchromatin. Yeah, that's it.
Yeah, great. And so basically to visual, let me just, I'm stopping the share for 1 minute. I want to pull out an image to show you people, which I always use to, it's an image molecular basis of inheritance. Wait. Where is it? Where is it? Yes. So I'll just copy this. Perfect. Yeah. Can you see the screen that I'm sharing again? Yep. No.
Yes, sir.
Yeah. So look here. So it's a very good illustration I got from Longman's Benjamin Cummins book. Yes. How are you, Parker? So we are. I think you left in between because of network issues. So we are doing. We are just revising what we did in the last class, which was we learned about DNA and how DNA, a very long molecule of DNA, is packed inside a very, very compact nucleus. So currently I'll be talking for another 5 minutes on DNA compaction using this figure. And then we will go to the search of genetic material, how actually, scientists figured out that DNA is the genetic material. Okay, is that clear, Parker? Thanks so much. I can see some.
Yes, sir.
Okay, great. So before we go on that search, let me show you this image because it makes things very clear. So let's start from here. Can you see that this is the one is a DNA double helix, right? And the ladder that we were talking about, the distance between the backbones, the distance that needs to be constant. Remember, we did a question that why a purine always base pairs with a pyrimidine to keep the distance constant. That constant distance is around 2 nanometers. Okay. And then this is one pitch. From this turn to this turn is one pitch. This illustration does not do justification because we know that in one pitch there should be 10 base pair stacks, but it is not the case here. But you know that in one pitch there has to be 10 base pair stacks, and the one pitch is 3.4 nanometer, right? And then this DNA, with the help of these histone proteins, gets wrapped. So now you see a nucleosome here, right? So can you see that a lot of DNA got packed? It's like packing, and then see these nucleosomes also get again packed like this. See here what I'm drawing. So this is now called a supercoiled chromatin. So this is chromatin number 2, and then this is a tight helical fiber, which is a supercoiled chromatin. And then this supercoiled chromatin also, it's like, I think how we make braids of hair, right? Long hair, we just keep coiling, and sometimes we super coil. So if you coil hair and then you super coil hair, you will be able to compact a lot of hair in a very, very tight space called the bun on the top of the head, right? You all must be more familiar with it than I am. But that's a very good analogy to understand. So it's supercoiled, then this supercoiled structure is again supercoiled. So you can see how many layers of coiling we see in this illustration is one here, where we are making nucleosome. So making nucleosomes is the first layer of coiling, sorry, condensing. Then this condensed thing is again supercoiled. So we see second here, where the tight helical fibers are made. You don't have to remember these names. This image is from a very advanced syllabus book. And then I'm just showing you to give you a context. And then again, it coils for the third time to make this fiber. And you can see that this again goes, and this is what forms what I'm forming here. What do you think? Now you'll be able to think. What is it? It's a chromosome, right? So this is how chromosome is formed after around 4 layers. So fourth is where, so you can see that there are 4 layers of compacting, and then it becomes a chromosome. So chromosome is a super dense and compact form of DNA. That's how a meter-long DNA can stay in a very, very small space. Okay, is that clear, everyone? And this linker DNA is what I was talking about here. So the DNA between the two nucleosomes is called linker DNA. Okay. If you do not degrade these proteins, but still give DNA degrading enzymes, you will see the beads will break, like bead on string structure will break into beads, because this part of DNA is still open. It always stays open, and it can always be degraded by the DNA degrading enzymes. Okay, so the linker DNA is what is left between two nucleosomes. Is it clear, everyone? So do you get some kind of a mental map for this whole process? Perfect. Now let's start today's class by asking a question. So today in 2023, because we know a lot about DNA genetics, things looks like we always knew it, right? We study the same thing that our elder brothers and sisters studied, and probably what our parents also studied, some of it when they were in their bachelor's and masters. So it looks like, oh, we always knew it. But that was not the case, right? At one point of time, Watson and Crick both were very clueless. They knew nothing, and they were just, discussing things with friends and pubs and clubs and coffees and dinners. And then we got to know that DNA is double helical. So, but long before we know about DNA, long before we know about anything, how genetics work, let's go back to the time of Mendel. Let's go to the last chapter. When Mendel did all of that experiment with pea plant, did he know anything about chromosome, gene? No, right? He did not know anything what's inside the nucleus and how is it getting inherited. In fact, he did not know that there's a term called gene or allele. This is how we study Mendel's work now. So that's where I believe that when we say these terms, that Mendel gave these laws, and in that statement, there is a word called allele or gene, we always think that, oh, Mendel must be knowing it, but he did not. We put some extra words later on in the syllabus. The scientists were very puzzled that what could be the genetic material. So Mendel proved something, and now everyone was puzzled that, okay, if there is something that passes on from father to offspring or mother to offspring, what is that thing? Is that a real thing? So people started going smaller and smaller and smaller into fundamentals. So if I break a body, I will go to organ system. If I break a organ system, I will go to individual organs. If I break an organ, I will go to what? Tissues, multiple tissues in that organ. If I break the tissues, I will go to individual cells. And that's where the limit was back then. So people started asking what is inside the cell. So they figured out that there is lots of things inside the cell: proteins, carbohydrates, lipids, different type of sugars that come under carbohydrates. They also knew that there are something acidic because I told you about nuclein, right? Frederick Miescher was the one who stained the nuclein. He discovered that there is something acidic in the center of the cell, and it is called nuclein. And then people figured out, oh, there is some acidic molecule also inside the cell, because when we do these chemistry from cell extracts, we get the hint that all these molecules are present inside cell. So then the quest started that, okay, we know what is the building block of cell. It is protein. We also know that what cell, like bacteria, needs to survive, like a sugar, whether it's lactose, glucose, sucrose. So what could be a genetic material? And if you or any one of you were a scientist back then, you would also have thought, I think it's protein, because protein makes the whole cell. Protein makes everything happen inside the cell. Proteins are enzymes. Remember, we talked about some functions of protein back up. Where did we do that? Here, right? Because in the central dogma, protein comes at the very end, and that is what is important for everything to happen in the cell. So protein forms the cytoskeleton, which is the structure. Proteins forms the enzymes, which helps in metabolism. Protein forms the receptors, which help a cell communicate with another cell. So many things protein does. I think protein is also the genetic material. That is what scientists' first guess was. But thinking is not science. Thinking is the first step towards it, right? You have to prove things in science. If you think protein is a genetic material, prove it. So let's go to the first person who tried to do something very daunting. His name was Griffith. So first thing that we are going to talk about today is Griffith's transforming principle. And let me tell you, we did not get to know that DNA is the genetic material just by one experiment or one person or even in one decade. It took a lot of time, so many people coming one after the other, doing something innovative. Then there were some loopholes in it. Then the next person came, did something more innovative. And finally, over a series of experiments, the scientific community was convinced that, okay, DNA is the genetic material, okay, and not protein or sugar or lipid or anything else in the body. So Griffith's transforming principle talked nothing about DNA at all, but still it is the first most crucial step towards that direction, like towards finding DNA as the genetic material. Now, what Frederick Griffith did, he used bacteria, which is Streptococcus pneumonia. So the model system was Streptococcus pneumonia. You know something about this bacteria already from disease chapter, last class 11th. You studied some human health and diseases. Was it in 11th or no? It's going to come. Okay, I sometimes get confused, because in my head everything is connected to everything. So yeah, sorry, but you must have heard the word pneumonia at least. Yeah, what is it? What is Streptococcus pneumonia like? There's a disease named after it because this bacteria caused that disease, pneumonia. What is pneumonia? Anyone knows pneumonia? No one knows pneumonia? Okay, that's very interesting because so if you have been raised in Saudi, probably you might not, because it's a arid and dry country. So pneumonia is more it occurs mostly in these tropical countries, especially countries which are developing and do not have a very high degree of water sanitization and things. So pneumonia affects the lungs, and it's caused by this bacteria, Streptococcus pneumoniae, and also another one called Haemophilus influenzae, right? These two names you will study later on. But remember that this, and what does strepto and coccus means? The coccus tells you about the shape of the bacteria. That I think you know already. So the word, for example, lactobacillus. So bacteria are of many different shapes. Sometimes their name tells about their shape, like lactobacillus, the bacilli shape, rod shape. Streptococcus, the cocci shaped, comma shape. So Streptococcus pneumoniae, he used Griffith. And the interesting thing about this bacteria is that it has two strains. Okay, it has a S strain and it has a R strain. The same bacteria has two strains. Okay, sorry, I should just write strain here, no S. Now this S strain is called S because the S word is for smooth. This bacteria, this strain of, so the species is same, it is the same bacteria, just like humans. So you can say that some humans are taller than some humans. So some humans, some races, I should not say races, but some people living in some regions have a very, very different physiology than some other people living in other regions. But they are same species, right? We are all humans, but some human are taller than the other. Some human are darker than the other. Similarly, this species of bacteria has two strains. The S strain form smooth colonies. So if you look at the colonies, what do I mean by colony is we always culture bacteria on these bacterial plates, and bacteria grow like this. So if you look at a plate, you will only see these kind of spots. These are called colonies, okay? Bacterial colonies. Now, some bacterial colonies are smooth and shiny, and the streptococcus that form smooth and shiny colonies are called the S strain streptococcus. And the other one is called the R strain for rough colonies, okay? So R strain forms the rough colonies. Now, first you have to understand why it happens. It happens because there is a gene which is functional in the S colonies, the S strain, which codes for a mucus. So S strain produce something that R strain cannot. Just like a dark-skinned human can produce melanin in the skin more than a white or a fair-skinned human, right? Similarly, this S strain bacteria produces mucus. Mucus is basically nothing but polysaccharide. Okay, it's a polysaccharide. So it produces a polysaccharide that coats over the bacteria and gives the bacteria a shiny look. R strain cannot produce that mucus or polysaccharide, hence it remains rough. The colonies look rough and non-shiny. Also, S strain is lethal. Okay, it can infect, or we use the word virulent. It can cause infection. Okay, but if R strain goes into the body, it is non-lethal or non-virulent and rough and non-shiny. Now, clearly, it tells us that something related to the virulence of the bacteria or the lethality of the bacteria is associated with the ability of the bacteria to produce mucus, correct? And that's true. So S strain infects through producing mucus and making a coat around it so that it can evade immune system and it can be more robust and cause infection in the body. Okay, so Griffith knew this, good knowledge. Now Griffith thought, what should I do with this knowledge? And since he was a thinker, he thought, okay, let me try something. So if I inject S strain bacteria into a mouse, it's very cruel, but that's what we have to do for betterment of humanity. Something needs to be sacrificed in science, and we decided it should not be humans. So let's go to animals which are closer and related to humans, and which can give us a lot of knowledge, and we can make human lives better and make medicines and all. So S strain, so he took S strain and injected in mice. Now, what do you expect the outcome to be? Simple. If you take S strain, you know it's virulent, you inject it into mice, what will happen?
Mice die.
Mice will die. Sorry for this poor Stuart Little, who sacrificed his life for science. Thank you so much. Not just one, many, but they all die. Then Griffith is like, okay, let's take just the R strain now and inject it into the mice, because anything that's told to you in the books, it might be wrong, right? So he just tested it, injected in mice, and what happened? What do you expect?
Mice live.
You will expect that the mice live. And that's true. Mice lived. Hooray. Yay, great. Because it was not causing virulence or infection. Now, then he did one thing. He said, okay, how can I prevent infection? So whenever a person is infected in your home, for example, Alia was suffering from some sore throat. If Alia is here. Yes, Alia is here. So Alia, did your doctor tell you to drink sanitized or boiled water or RO water, and take care? Do not eat anything contaminated. Do not eat anything from outside. All these are standing advices, but why are we given these advices? Especially about boiled water. What does boiling the water does?
I think it kills the bacteria which are present there.
Exactly, you think correct. So when we boil the water, anything, not just bacteria, anything which might be present in the water, and water is a reservoir for sustaining life. Lots of life can sustain in water, just in water, right? You don't even have to add a lot of things into it. So even if it is just a tap water, there could be bacteria in it, there could be fungal spores in it, there could be viruses in it. And most of these can be killed simply by boiling the water, because they cannot sustain that higher temperature. Okay, perfect. So that's why we kind of sterilize the water. So he also did the same thing. He took S strain and boiled it. So if you take S strain and boil it, you expect the bacteria to die, right? Now we call these bacteria as heat killed. Because remember, long ago, before this, there was another person who told us, let me tell you how to store milk. He told us that pasteurize the milk. His name was Louis Pasteur. And in his name, we call the process pasteurization, where all the milk that come to your home, which is tetra packed or in poly bags, it's basically heated up, very slow heating. It goes up till 60, 70. Bacteria die in it. And then it's cooled down again, then again heated up and again cooled down and then packed. So that's called pasteurized milk. So heating kills bacteria, we know it. That's why we also cook the food, because anything that we're eating, especially non-vegetarian food, because it's a tissue, it's a living cell, not just non-vegetarian, but even vegetarian food has a lot of bacteria in it, but those are plant pathogens. And we are less affected by plant pathogens, but we can be more affected by animal pathogens. So it's more important to cook flesh properly, but vegetables you can also eat raw. Okay. So when he took the S strain and heat killed it, and then again injected in the mouse, what do you expect, Alia?
He lives.
Not he, the
Sorry,
mice.
yeah, sorry, nice, yeah.
Yes. He was not doing it on himself or the humans. Though, there is one person who was so desperate in science, he actually did an experiment. It was the first human experiment, but there were no rules back then. So he did the experiment on humans, even his own son. Anyone knows the name of that person? He was an amazing scientist and thinker who almost, put his own son's life on stake but he was very confident on his logic and science that my son will live. He injected a virus in his own son's body. Do you remember that person who discovered vaccination? Edward Jenner, right? He discovered the first vaccine for smallpox, sorry, cowpox. Cowpox was a thing and smallpox was a thing back then. People used to die in like millions because of these two poxes. We have a very mild version of it called chicken pox in our time. If I don't know if any one of you have had chicken pox, because we get vaccinated for it very early. But when I was very little, I got chicken pox once. And when you get that, after that, you are naturally immunized for the virus because your body fights it and makes antibodies. So Edward Jenner actually injected, he'd killed these pox viruses inside people's body, including his own son, and proved that nothing will happen because you are now immunized. You have immunity against it. So people have done all sorts of things in science. Okay, that's how we live today. But now we have strict rules to not do these things on humans. Okay, because science is very open. Everyone can do science, but you would not like everyone going and injecting things in everyone's body. Okay, so it needs to go through some protocol. So when he took S strain, heat killed, injected in mice lives. Till here, everything was going as per logic, right? Makes sense. Now he got an idea, and that idea changed the thought process of scientists from that point onwards. Now look at these two things, people. When we look at experiment 2 and experiment 3, what is the common thing between these two? When you inject a R strain, and remember that this R strain here is a live strain, and this S strain here is a heat killed strain. But when you inject both of them, what happens? In both the cases, mice lives, correct? Yep. Now let's go to the most interesting part. So he thought, okay, it makes all the sense, but let me just give it a try. That's what scientists do. Sorry, what happened? They just give it a try because of, they think something. So he thought, what if I make a cocktail of R strain? I take a R strain, which is live, and add it with S strain, mix it with S strain bacteria, which is heat killed. Now, when you did those experiments individually, in both the cases, mice lived. But when he injected this, one would have expected the mice to live, right? Yes or no? If I would have asked you, what do you think will happen if I inject these two together? What will you say? And this is exactly what Griffith also thought, by the way. It's not like you have to say, I think the mice will die now because something, that's how, if the mice lives, it is very anticlimactic, right? That's why you're teaching me this. NCERT says the mice lives, but yes. So in this case, the mice should have lived, but instead, when he injected this, the mice died. And he was very puzzled. So he thought I did something wrong. He thought maybe the S strain was not properly heat killed. He repeated the experiment. This time he boiled to even higher temperature. Again, S strain boiled, all bacteria dead, mixed with R strain, injected, mice died. He was again puzzled. Then he thought, no. Maybe my R strain is contaminated already with that strain. So he started with a new culture of R strain, heat killed S strain again using a very high temperature, then again injected, mice died. Now, by this point, he was convinced that something is going on here. The mice should have lived because when I was injecting them separately, the mice was living happily. What is happening when I'm injecting them together? What do you think could happen? What is your logic? Aisha, what will you logically think? See, if you understood this experiment, you will never have to remember it. Like I have just told this so many times to students that, it feels like I did it in the past life. It's like that. So yes. So what do you think, Aisha? What is happening here? Why did the mice die? Give me all your hypotheses. What could have happened? He took care of everything. R strain was not contaminated. That is one thing that could have happened. He checked for that. S strain would not have died properly. Some bacteria must still be alive. He checked for that also. He increased temperatures and went to a point where he was sure that no bacteria would have survived. And what is the way to test that your boiling is good? You just inject a single S strain, which is heat killed, and he saw that the mice lives. So he was convinced that the strain is dead, heat killed. That's why when I'm injecting it alone, the mice is living. That's called a positive control. Sorry, that's called a, yes, in this case, a positive control where we know that the mice will live. But every time when he's injecting it together, the mice is dying. So what all could happen? Come on, people. Yes, Serum.
Maybe
I
something
know
about
that Iram.
transfer.
Yes, I know Iram knows the answer. She's just trying to sugarcoat so that she looks like, I just I know that I know that you're thinking right. It's your own thinking. I'm not saying that. Yes, just complete your answer. That's the beauty of discussions. Yes. And Iran's trying her level best to pretend. Maybe something got. I don't know. Maybe that's yeah. Yes. So what happened? Tell us. Something got
This
transferred.
is
You are
something got
perfectly correct.
transferred from yes, sir.
Yes.
Transferred from the S strain to R strain, or maybe R strain to
S.
S strain.
Something got transferred. Okay, that's where it tells that Iram thought it genuinely. So because if she would have just given the answer from NCRT, she would have given it 100% correct. So she says that something is transferring either from R strain to S strain or S strain to R strain. Now thinking logically, S strain is dead, right? So if anything gets transferred from R strain to S strain, it cannot revive a dead bacteria. There is one truth in biology, and that truth is once dead, dead. That's called death. Death is the final, ultimate truth of biology. So if an organism dies, it cannot resurrect from dead. A new organism can come back and start growing from the spores. That's a different thing, but not the same organism. So if S strain is dead, heat killed, anything that comes from R strain will not be able to revive it, right? Do you understand that? But that was one hypothesis they actually tested. NCRT does not tell you about this, but what Iram is thinking exactly what came in Griffith's mind. And he thought, oh my goodness, did I just figured out a miracle where a dead bacteria revives back? But he was disappointed that no, it's not that dead bacteria. So let's remove this branch for now. So clearly there was something getting transferred from the S strain into the R strain, which was making R strain lethal and virulent, where it should not have been lethal and virulent, because this is the only living thing which we are injecting, right? And then he calls that transfer as the transfer of genetic material. Okay. Transfer of some material which might be genetic that transforms a non-virulent strain into a virulent strain. Okay. Transfer of some material genetic that transformed. So this is called transformation. That transformed R strain to S strain. So R strain became S strain by getting something from the S strain. And now it won't be very difficult for you to think what was transferred. So today, now we come back to 2023 and you can answer, I know what got transferred. The gene that codes for that polysaccharide code got transferred, and all of a sudden R strain bacteria became S strain bacteria. Yes, right? You're thinking that? If you're thinking that, you're thinking correct. That's what happened. But let's go back now. Okay. Griffith didn't know this. So he just said that something is transferring and which is transforming. Now, what is the way to prove it that there is real transformation happening? This is just a hypothesis. How can you prove that, yes, a R strain really got transformed into S strain? Can you think of a way? So I am injecting live R strain mixed with dead S strain into a mice and mice died. How can I prove that R strain really got transformed into S strain by getting some genetic material? Can you think of a way? Anyone? Mariam? Mariam just joined us. She joined long back, I guess. Mariam, are you there? Can you, yes, she's there. If you were Griffith, what you would have done to prove that real transformation of R strain into S strain is happening? Parker? Come on, at least this thing, you people know it. You know, I know that you people know it. I don't know why you don't speak.
So
Am I?
can I?
Yes, just 1 second. I just want to, infuse the same enthusiasm into others so that this class can become a very happening place. You know, am I like too boring when I teach, or too intimidating? Like you're scared of me? Like I'll just scold you for giving a wrong answer. That how dare you give a wrong answer in my class? I cannot even say get out of my class, because how can I say get out? You cannot get out, right? Come on. Yes, Iram, what do you think? How will you prove it that the real transformation happened?
I just forgot it. So sorry, can you repeat?
Yeah, so my question was, I took R strain live bacteria, mixed it with S strain dead bacteria, injected it in mice died. And then I am assuming that something is getting transferred from S strain dead bacteria to R strain live bacteria and transforming the R strain live bacteria into S strain bacteria. Now, how should I prove all of this?
So when you just take off the S strain, then there is no transfer of anything, right? But when you have the S strain, we know that definitely it's from the S strain that something is getting transferred to us.
Right, you're perfectly right that when I'm injecting just the R strain without a heat killed S strain, the mice is still living. So the only thing which can do something is S strain. But still it is behind a curtain, right? How will I take off the curtain and show to the world that, see, this is a real proof of my hypothesis? Because in science, many things make logic, but we do not accept it all of a sudden. That's what the beauty of science is, that any word coming from anyone's mouth is not the truth unless and until it is tested, right? So I can think of one way, and you people tell me that if that's correct or not. So this whole point of showing that a bacteria is R strain and S strain, let's go back, is to show a colony, a cultured colony of that bacteria, and show that, okay, they are growing in shiny colonies, so they are S strain, right? And they are virulent. And they are growing in rough colonies, so they are R strain and they are not virulent. So what Griffith did, and what is the only way to prove it, is when you inject both of them together and the mice died, then you take a tissue sample from the mouse body, make an extract of it, and isolate bacteria from that sample. Now remember, the only live thing which went inside the body was R strain. But when you isolated the bacteria back and put it on the culture plate, when they will start growing, if they start growing in smooth colonies, which means they are living and they are S strain. So how can a living S strain come out of a mice where no living S strain was injected? A dead S strain was injected and a living R strain was injected. But what came out of the mouse body is a living S strain bacteria. Is it a proof enough to say that, see, it got transformed inside? And that's how I can get back live S strain bacteria? Because the other option of a dead bacteria becoming alive is not viable. Okay, once dead is dead. So that's what Griffith did. So write down. He cultured successfully live S strain bacteria. He cultured successfully live. He isolated and cultured. Sorry, write the term isolated. That's very important. He isolated and cultured successfully live S strain bacteria from the dead mouse tissues. From the dead mouse tissues. Which was injected by a cocktail of live R strain and heat killed S strain. Live R strain and heat killed S strain. Now that's amazing, right? Okay. And this, so he proved his transforming principle. So this was all good. He became a celebrity. Everyone, was like, wow, great job, well done, woohoo. But still no idea about what is getting transferred. Still, we don't know what is the genetic material, right? So what can we do to find out what is the genetic material? So he opened a doorway, and before Griffith could think and do this, the next step, he could take the next step. I'm sure that if you would have given Griffith some more time, he would have thought the experiment and did it. But as we say in science, the beautiful thing is I do step one, I solve step one, and then Iram can quickly come and take inspiration and understand that step one better than me myself and do step two, and that will take the research further. And then someone comes in and completes it by doing step three. How many of you know about Einstein? Albert Einstein, the most celebrated scientist because of his personality, and everyone has seen his tongue out image, right? Where his tongue is out and his hair are all fried like he got electric shock. Anyway, so, but how many of you know what Einstein got Nobel for? Einstein got a Nobel Prize, right? But do you know what Einstein got? What is Einstein's best work of his life? What do we know Einstein for? He's known for theory of relativity, right? The
Yes.
general theory of relativity and the special theory of relativity. And that because of theory of relativity, now we are able to understand cosmos, space, and everything. So that's not just a theory. Now we have proofs and practical things for it. But he did not get Nobel for the best work that came out of his mind. He got Nobel for explaining something that was done by someone else. He got Nobel for explaining photoelectric effect. Okay. And who was the one who figured out photoelectric effect? I think that you must have done in your physics. And let me tell you, all of you, Mariam, Aisha, Alia, Iram, and Parker here, if you really want to become a very good doctor and you want to go to a very good grad medical school, you need to have a very good physics and a very good chemistry. Biology alone can only take you so far. It cannot it cannot make you. So 50% of the syllabus in NEET is biology, but just know that that 50%, most of the people who want to become a doctor are good in biology. That's why they want to become a doctor, because they have an inclination towards life sciences, living systems, animals, humans, so on, diseases, etc. But what we are not good at as biology students sometimes is mathematics. I was also not very good at mathematics when I was in my high school and in my undergrad. And that is what, prevented me from becoming a doctor. Also, I was not, after one point of time, I did not wanted to become a doctor, to be honest. It's not because, it's not like the story of the fox that, oh, you could not get the grapes, that's why the grapes are sour. It's not like that. I really lost the nick for it. But what I always advise, but I realized, I thought that, okay, my maths is not good. My physics was very, very good. I really liked physics. So biology was my first favorite and physics was my second favorite, which is very, very, it's a little, it's, it's not natural, right? You will think that, okay, you are a biology student, you like biology a lot, then the second favorite must be chemistry because we have a biochemistry, organic chemistry. But for me, it was opposite. I was very, very weak. I'll not say very weak, but clearly weaker than you people in chemistry. And my physics and biology was very, very good. My mathematics was also again weak. Then I thought, okay, I can become a researcher and a scientist, and I don't have to do mathematics again. And then I was proven wrong when I joined PhD because I realized, oh, no, no, no. You cannot do anything in the world without mathematics. Let me tell you. It's very true that mathematics is called the queen of all sciences. Okay. It's the purest form of explaining anything which happens in biology, chemistry, physics. Because without quantifying, you cannot do anything. You cannot even treat a disease. You cannot even treat fever. Because to treat fever, you have to know how much drug needs to go in the body to treat the fever. If you give more than that drug, the body will go into a hypothermic shock and the person will die with the same drug. Okay. So again, mathematics. You have to quantify receptors, again, mathematics. Okay. So please work on your physics, mathematics, and chemistry alike, like you are interested in biology. Once you are into the med grad school, then you will have some breathing space from maths, physics for some time, and maybe for forever also. I don't know. Okay. But even after becoming doctor, if you want to come into research, you will have to come with all your gears of maths and chemistry back. Okay. So where were we? Where did it start? Yes. Yeah. So before Griffith could think of this experiment, there were three people who independently were reading Griffith's work. They were very, very, kind of motivated by Griffith's work. And they thought, okay, so what our fellow person did is that he showed that something goes from one type of strain to another type of strain. So they thought, let's characterize it using chemistry. So biology cannot give us all the answers. Let's go to chemistry. So they did, for the first time, the biochemical analysis. Okay, that's called the biochemical characterization of the transforming principle. So they actually did the second step of Griffith's experiment only. They decided we will repeat the same experiment. So they repeated the same experiment. And when R strain got converted into S strain and the mice died, after that, let me first name these three people. So they are, first is Avery. His name was Oswald Avery, but if you just know, oh, it's a V, I think. Avery, Oswald. I messed it up here. Oswald Avery, Colin MacLeod, and it was some McCarty, but what was the first name I forgot, but it was some M. McCarty. sorry, no, A. I'm sorry, I forgot the first name. McCarthy. So often in the books, you will find every MacLeod and McCarthy's experiment. So three of them. Yeah, someone is helping me. Yes, Mariam is coming to the rescue. She says the first name was Macklin. Okay, yes, right. Sounds familiar to me. Though I'm not very sure if it is the right answer, but I think it's the right answer. So Macklin McCarthy, it also goes, it rings, it has a ring. So Macklin McCarthy, Colin MacLeod, and Oswald Avery, they did biochemical analysis of the transforming principle. So what they did, they purified chemicals from the S strain, because that is what the idea is, right? Something from the S strain is going to R strain. So the purified chemicals from heat killed S strain. They did repeated exactly the same thing, but this time when they heat killed, after that, they purified chemical from heat killed S strain Streptococcus pneumonia. Now, it is a very, very daunting task. It's a very tedious task. When you purify chemicals, there are all sorts of chemicals present inside the cell, right? So when you talk about chemicals, what all things you will get? You will get proteins. So there'll be a lot of proteins, all different kinds of protein, but it did not matter what protein it was. It should be just falling into the category of protein. So they purified proteins, they purified lipids, they purified all sorts of um, saccharides or sugars, but I'm writing saccharides. Just one second, people. Yeah. Saccharides, short is sugars. And they also purified nucleic acids from the nuclein, from the nucleus, right? And now today we know that nucleic acids are of two types. One is DNA, deoxyribonucleic acid, and the other is RNA. So in your books, you will find written that they purified DNA and RNA, but they did not know about it at that back then. So they purified nucleic acids. So all these are different kind of molecules. Now, what is the next logical step? Simple. One by one, take these purified chemicals that came out of the S strain and inject it into mice. Along with what? Along with the live R strain. Okay. Hiram, you are mute if you're speaking. That's why I could not hear you. So they took proteins from the S strain, mixed it with the R strain, and injected it. And when they injected protein, what happened? What happened? Nothing. The mice lived. So if the mice live, this is one way to do it. Another way to do it is you use enzymes that degrade these things. Okay. So if you cannot be 100% sure, like doing this way, one can say that, okay, what you say as proteins maybe is also contaminated by nucleic acids. How are you sure that it is pure protein? So they also did this one thing. Apart from injecting in mice, what your NCRT tells you is that they use digesting enzymes. Digesting enzymes. So these are the enzymes. So here the work becomes little easier. You take the extract from the S strain, heat killed. Then inside the extract, if you put protease. So if you take a protease, which is an enzyme, what will it degrade?
Protein.
Yes, it degrades proteins. Then you take lipase, it will degrade lipids. Then you take, for sugars, there are many different kind of enzymes, amylase, all these like sugar digesting enzymes. You take that and it digests all the sugars. And you take nucleases, and it digests all the nucleic acids. So they found that the protein digesting enzymes and RNA digesting enzymes and lipid digesting enzymes and sugar digesting enzymes, all of them did not affect the transformation. Okay, so the transformation was happening normally. So when you break down protein, still R strain is getting transformed into S strain. When you break down all the lipids, still R strain is getting transformed into S strain. When you degrade all the sugars, still R strain is getting converted into S strain. When you degrade all the RNAs by using specific RNases, still it is happening. But when they use DNases, deoxyribonucleic acids degrading enzymes, then the transformation did not happen. Now, does this tells you something? Only when you use DNases, the enzymes that degrade DNA, there is no transformation happening. So what does this tell you? That DNA is the material that is getting transferred from one strain to another. Does it make sense, everyone? I
Yes,
want a
sir.
thumbs up if everyone understands this experiment. I just got 2 thumbs up. What does this mean? 3, 4. Okay, I'm just getting it slowly, but I'm getting it. Parker, did you understand the experiment? I did not get any thumbs up from you. We leave no one behind. Okay, so now write down. Oh, Parker just got disconnected. So could you not understand the full experiment, Parker? Yes, no? No, okay. So Iram, how about you give a quick gist of what every McLeod-McCarthy did to your fellow classmate? When learning comes from peers, it is the best. And then I will also feel good. And I can drink water in the meantime. Iram, are you there? Can you help? Or Alia can also help if she wants.
Sure. Um,
Yeah,
so we
great.
were doing, so after transforming principle, we didn't get how there were some answers which were left unanswered. So three scientists, uh, Avery, McCartney, and McLeod, they purified some biochemicals from heat killed estrane.
Yeah,
And
they
those
purified
biochemicals,
all the biochemicals.
yes,
They
so
did not leave anything behind. They purified, basically they made an extract from the S strain, a soup, a S strain soup without any masala. Yeah, that's called the
so
extract. Yeah, then
uh, so then they use some digesting enzymes like protease or lipase. So these digesting enzymes digest these biochemicals. For example, protease digests the protein, and lipase digests the lipids. Um,
perfect.
so when when they injected the protease in that, um, it didn't affect the transformation. Like the transformation still happened, and the mice still died. So then, they did same with lipids and so on, digesting enzymes. Then, when they did with DNAs, the DNA gets digested, and the transformation didn't happen.
Did
And
not happen.
yes,
Yes, perfect.
so
Very good. So what does this tell you?
hence the DNA is the transforming
Material.
agent material. Yeah.
Yes, that's great. Parker, could you understand what Alia said? It's like they got rid of one component at a time by using digesting enzymes. They first removed proteins, Still, R strain was transforming into S strain. Then they removed lipids, RNA, sugar, still it happened. But when they removed DNA specifically, but other things, so when they are removing one thing, the rest of the other things are there in the in that chemical soup. But when they just removed DNA and rest everything else was there, no transformation happened. And that's how Alia is concluding. Yes, Parker says yes, she understood, which means Alia is an amazing teacher and explainer. Well done, Alia. So that's where Alia is concluding that she is convinced what every MacLeod and McCarthy did. She's convinced that DNA is the genetic material. But sadly for these three scientists, all the community of science was not convinced. Now you will feel bad. You're like, what? So much of hard work they did, and still people are saying that we are not convinced. But yes, that's what science is. Science is very robust. So why were people not convinced is because first, at that time, they were not 100% sure that protease, one kind of protease can degrade all proteins in the cell. Today, we know that it's not the case. So back then also, people were a little skeptical that what they did is okay, but maybe they left some proteins, maybe they left some lipases, maybe they left some DNA when they did this digestion. So we can't just conclude that DNA is the genetic material. But also there was one school of thought that was convinced, just like Alia. But in biology, we still do not give them the credit which they should get. Even NCERT does not give them the credit to prove. But according to me also, they were the first people who proved that DNA is the genetic material, but still with some loopholes. Okay, Alia, do you agree with me? We both are on their side, but we also understand that there were some loopholes, right? Maybe the protease was not pure. Maybe the lipase was not 100% effective. Do you understand, Alia?
Yes, sir.
Yeah. So back then also people were not very convinced. And then came one of the experiments that gave the unequivocal proof, like undisputable proof that yes, DNA is the genetic material. So the next step was taken by two scientists called Alfred Hershey and Martha Chase. So this duo did something so fantastic that it became the unequivocal proof in biology. And it actually, it took, so you must be thinking that, let me tell you something. So when these three scientists worked, it was a very, very long time. It almost took, if I'm not wrong, a whole decade, somewhere around 1930s, early 1930s, maybe 32 or 33, till 1943 or 1944, something like that. It took 10, 11 years for them to independently work, do all this. You just studied this in 10 minutes, right? 20 minutes. It took them 10 years to do all of this and prove that DNA is the genetic material. They were convinced, their supporters were convinced, people who, the school of thought that was in their support was convinced, but the whole community was not convinced. People found some loopholes, and those loopholes needed to be answered. They also tried to answer those loopholes through their biochemical analysis. But again, when biology could not do it, one step further chemistry took. But when chemistry also could not solve, then two scientists came. And from here, they came somewhere around in 1950s, again, another 10 year, let's say around 1952. So another 10-year passed away, and then came Alfred Hershey and Martha Chase. And they used physics, okay, like biophysics, biochemistry, and biology, all three together to design an experiment that will give us 100% proof that DNA is the genetic material. Okay, actually, they were also, to some extent, in support of the experiments and the results of every MacLeod and McCarthy. That's why they wanted to prove it. So by this time, when they came, there were two candidates. Either it could be protein. The war was protein versus DNA. Okay. And they proved that DNA is the winner and DNA is the genetic material. But how they did this is something we will talk about tomorrow in detail. But let me set the stage for you today and give you the homework. So tell me, before we go into the experiment, tell me if you have to prove between two things, what logic will you use? So let's say me and Iram were invited by Alia at her house, okay, for a dinner. Then she kept one dessert bowl with just 2 scoops of ice cream in the center. She went away for some time and she came back and the bowl was empty. And now it's between, she has to figure out who, and we both are saying that, okay, we don't know who ate it. So it's between me and Iram. So whenever it's between two candidates, what logical criteria will Alia follow to exclude one or include one, something like that? Now you can very well say that, oh, Alia's common room had a CCTV and it's very clear. But that's not the fun part. If that happens, then I think I'll be the culprit. But you have to, whenever we have to choose between two things and exclude one and include the other, like black and white kind of an experiment, we choose characteristics which are very different in both. Okay. So Alia will take, first steps of Alia will be to figure out who among us, among me and Iram, is a bigger sweet tooth, who likes dessert more. So if Iram says that, okay, and here we are assuming that we will always, when Alia will question, we will say the truth. So if Iram says that, oh, I do not fancy ice creams at all, and I say that, yeah, I do like ice creams, then Alia will get one step closer to say that, okay, one characteristic that helps me to figure out between these two is their inclination towards this, or the flavor then. Okay. Similarly, Alfred and Martha, what they did, Hershey and Chase, what they did, they thought, first thing is, the world did not believe the above three scientists because they could not show things. When you see something in science, you believe it more. So they wanted to make it a visually a visual proof. We say just visual. They wanted to give a visual proof that which is a genetic material. Now to do that, by that time, there was a technology called radioactive labeling. So your first homework is, before you understand this part, you will go back today and read about radioactive labeling. It simply means you can use a radioactive isotope. You have studied about isotopes in physics. That will make my job easier if you have. Have you studied about isotopes already?
Yes,
in
sir.
atoms? Okay, perfect. So you know what are isotopes, right? Anyone who knows what is isotope? Again, see, you have to know physics. Come on. That also you figure out then. I'm giving you homework to understand radioactive labeling. And you can, using this technique, which you will figure out, label any element, any atom, any kind of atom. So they started looking for what is different between protein and DNA. So something that only protein contains and something that only DNA contains. You already know that carbon is present in both, right? Hydrogen is present in both, oxygen is present in both. And what is different? So one thing that is only present in DNA is phosphorus. It has a phosphate backbone, right? And one thing which is only present in protein is sulfur. There are many things present in protein which are not present in DNA, by the way, but they chose sulfur. So there is no sulfur present in any DNA structure. nucleotide, nucleoside, nowhere. Only proteins have sulfur because there are amino acids that contain sulfur. Cysteine contains sulfur. And DNA has phosphorus that proteins do not have. So they use this technique to radio labeled sulfur. So in one case, they labeled all the sulfur. In other case, they labeled all the phosphorus. And then what they did next is what we will talk about in the next class. And we will finally figure out how DNA was found. Then in the next class, only from DNA, we will move to RNA and we'll go to central dogma. Okay, after that, we will do replication, transcription, and translation. So please read this chapter from NCERT, people. Is the story till here clear to everyone? Anyone who's lagging behind, please let me know. If everyone is on the same page, give me a thumbs up. This is very important. Okay, perfect. So I'm getting all the thumbs up. Alia, Aisha, Iram, Mariam. Okay, perfect. And also Parker. Cool. So I'll see you in the next class. Remember the story. Go today and tell whatever we studied till now, this story of history of science to your parents, to your, your siblings over dinner. Just say that, okay, today I want to tell you something. Do you know how DNA was found out to be the genetic